C terminus in the lumen. So we saw, right, this here, right here. So it's N terminus in the lumen and it's C terminus in the cytosol. One way was to have the ERSS in the middle and then the stop transfer sequence. But if this N terminus is relatively short, we can do it another way. If the N terminus is short, and we're going to say just for the purpose of this course, because people like numbers, less than 50 amino acids, then another way to do it would be to have right here, let's move this up so that looks relatively short right here. So we have N, C, and that's a start transfer sequence. With a net number of positively charged amino acids right here. Okay, so we have two ways to do this if the N terminus is short. Question here.
Just for clarification, so you're basically saying that it might be coming out of the ribosome one way, but it will adjust however it needs to based on charges into the translocon?
Perfect, you got it, yeah. And why? Why do the charges matter? Remember that the environment of the ER is full of calcium, which is itself positively charged, and it'll tend to have a net repulsion for positively charged amino acids. Why does, for charges themselves, why does the size of the N terminus make a difference? And the reason it makes a difference is that if we have a pretty long N terminus right here, remember this is going on and folding and folding and folding and folding, and we have a pretty big folded structure right here. So when the start transfer sequence emerges, binds SRP and goes to the SRP receptor and goes to the translocon, even if the positive charges were on the C terminal side right here, this could not be forced through the translocon. So this is forced to go like this, regardless. If the N terminus is relatively large and folded, it'll only go like this, and we'll end up with the N terminus in the cytosol and the C terminus in the lumen of the ER. Is that clear? Cool. Okay, so when you're looking at a structure, when you're looking at what we would like you to do, and we have homeworks about this, and this is the kind of things we like to ask on exams, we might get is to be able to go both ways. So if we give you something like this with all the signals in it, we would like you to draw what the structure, the predicted structure of the membrane protein is. So you'll need to look at the size of the N terminus, the number of positive charges, and how they're oriented. Or we might give you something like this and want you to draw the structures to go both ways to show that you understand this. Okay? Question here.
In the case when we get something like what's on the right,
like this right here.
Yeah, how would you notate that the N terminus is short enough?
Okay, if we gave you the amino acid sequence, you could count right here, and then you'd look at the character of the amino acids right here and count the positive charges, or we might tell you how big this is. Okay? Other questions? Yeah?
If you have like a segment that was had a significant negative charge and not like a significant positive charge, would that act similarly to where the negative charge would face closer towards the lumen?
Correct. That's correct. So it's the net positive charge. So an increase in negative charge is like a decreased positive charge. Yeah. Other questions? Okay. So in office hours, we're going to have these ropes So feel free to come and we'll just do a bunch of these and we'll give you some problems and we can work problems. The way that anybody who's been to office hours so far knows that we have a large room and we just sit everybody around the table and then sort of go ask questions. Anybody who has questions, of course, we answer them. And we do it as a group because if you have a question, probably somebody else does. Feel free to come if you don't have questions because you'll probably be inspired by other people's questions. And you probably have some kind of questions, and we'll just work group exercises together. So come to office hours. It's best, of course, if you come prepared. So if you're way behind, you can still come and engage in it, but people who come without questions in their head or without having worked the problems get much less out of it. They end up just taking notes like in class, and we want it to be sort of it's best for you if it's sort of a working session. So our office hours are on Friday, and all the details are in the syllabus. Be sure to come. Okay, and when we change them, occasionally we have faculty meetings or something, and we always send an announcement out that we're not going to be there. But we've set aside that time for y'all, so.
It's actually this Friday.
Okay, I thought so. I couldn't remember if it was last Friday or this Friday.
This week we're changing the office hours to 1, starting at 1 instead of at 2:30.
Yeah, and we're in Patterson, so it's not Painter. It's P-A-T. Some people end up in Painter, but it's Patterson. Okay, so this one has the rules right here that you can follow, right here, the N-terminus and the C-terminus, and it's caused by the voltage gradient high calcium right here. So work through that yourself. And then, any questions? Yeah.
So we mentioned previously that the ERSS is preceded by positively charged amino acids, right?
Okay, not the ERSS. The ERSS is the shorter one. It's not going to end up being a transfer a transmembrane domain, so that's always going to be cleaved. But I think you mean the start transfer sequence. Okay, so continue your question, but use the word start transfer sequence, I think. Unless maybe you were talking about the ERS. Here, just ask your question.
Okay, so I think previously mentioned that the ERSS is preceded by positively
Oh, I
charged
got it.
amino
Yeah.
acids.
Yeah.
So I was just wondering if do those amino acids not affect orientation?
They probably do. In general, when you compare ERSSs, so I rephrased your question incorrectly. You had a good question. The ERSS is less than 20 because it's not going to be a transmembrane domain. It's going to be 10 to 15 or so non-charged, generally hydrophobic amino acids right here. And I told you that there are generally positive charges right here. And that probably does affect the orientation of that ERSS so that the cleavage site right there is exposed. I don't know anybody who's experimentally modified that to look and change those amino acids and see if that affects the orientation or not. But I imagine that it does. Good question. Other questions? Okay, let's go to our first, let's see, I'm going to go back to that. Bunch of movies, they're all on campus. Okay, now let's go to this clicker question right here, or this Instapoll question, our first one. The HSP 70 HSP 70 chaperone has the name BIP. Sounds like a fraternity guy, doesn't it? But it's not. It's historical reason. It's called binding protein that was bound in immunoglobulin proteins. History doesn't matter, but it's called BIP. So that is the ER chaperone. And now this ER chaperone is soluble, meaning that it's floating around the ER lumen. So I want you to think about and answer this Instapoll question by yourselves. I'll tell you why. So the amino acid sequence for the BIP gene does not have an ER signal sequence. That is incorrect. So someone who chose another answer, can they explain why they rejected A as a possibility? Yeah.
Well, if it lives within the ER lumen, it has to have this signal sequence to even be brought to the lumen.
Right. BIP is a protein. So, and it's soluble in the ER, which means that it must have this structure here. And the only way that a protein can get into the ER in the first place is to begin with an ERSS. And then it's a chicken and egg, but other BIPs, of course, had to fold this to get it into the proper orientation. So this is what, when I tell you that BIP is soluble in the ER, you know that it looks like this. Soluble means it's floating around, it's not tethered to the membrane. And it's in the ER, so it had to have an ERSS. So A is false. And this sort of true-false approach that we're going to go through answer by answer is a good strategy for taking multiple choice exam questions, is to go through and analyze for yourself whether they're true or false, and then go back to the true answer and see if that still makes sense. Okay, so the amino acid sequence of the BIP gene lacks an ATP binding site. And some people chose that, but that's in fact false. So can someone who rejected that answer explain why they thought that BIP must have an ATP binding site? Back here. Right, you knew that it was an ATPase, so in order to do that, to be an ATPase, it has to bind ATP in order to split it. Excellent, and you knew that because you were comparing it with HSP70, so it must be an ATPase, must bind ATP. And the ER lumen lacks ATP, so BIP functions differently from HSP70. Not sure so many people chose that one, but the ER has a lot of ATP in it. So the ATPase activity of ATP is a small soluble nucleotide. It goes in and out fine. So it's rich in the cytoplasm. It's rich in the ER. It's not limiting. There's ATPases all over. BIP does not bind to hydrophobic patches on other proteins. That one's also false. So can someone who rejected that answer explain why they rejected that answer? Go ahead.
Well, I was thinking that if it was like HSP70 and HSP70 binds to hydrophobic patches, then it would act in the same similar way.
Right. What you're remembering is how HSP70 works. And so from Dr. de Lausanne's lecture, so as the protein is being synthesized, hydrophobic patches are exposed and HSP70 binds onto those. And the same is true in this ER lumen. As the N-terminus, or as proteins experience the environment of the ER lumen, either a soluble one or the portion that's in the ER lumen, as it's emerging, these HSP70 is binding to it. And because it's binding to the portion that is in the, so let's bring this to the cytoplasm, put it in here. So this is the lumen right here. As HSP70, a bunch of HSP70s are binding right here, they actually serve sort of like a wench, sort of like a binding thing. As they're folding this, they are actually helping draw the protein into the ER lumen. So, and it does so, the binding site for HSP70 or BIP are hydrophobic patches that are exposed as the protein is being, it helps the folding because this in the tertiary structure, a hydrophobic patch down here might need to bind with a hydrophobic patch down here, and so this needs to sort of wait and stay there until it's ready to bind into the appropriate tertiary structure, which might be relatively complex, and not right next to each other in the primary structure. Good, does that make sense? Okay, so it does bind to hydrophobic patches, and